Assalamu alaikum. I am Shiza and the topic that I am going to present is polymer microarray technology for stem cell engineering. Before delving into our topic, I would like to explain briefly what microarray technology actually is. Now, as the name indicates, it is a kind of technology that incorporates something like microarray. Microarray terms here suggest that it is an arrangement of something in rectangular form. So we can also say it as in the form of chips. So microarray technology is like a powerful tool that helps scientists study many things at once. It's like having a bunch of tiny spots immobilized on a surface and each spot contains a different sample. Figure here makes my point clear. By putting cells or molecules on these spots, which are shown somewhere here, researchers can see how they interact and learn a lot of information quickly. It's kind of like doing many experiments all at once, which saves time and gives us a better understanding of how things work. The second figure gives more insight via different steps. As we can see, there are the steps written here. So here, RNA is extracted from the samples and then reverse transcribed in the form of complementary DNA, which then hybridize with the samples. Now, the complementary DNA here is illustrated by these green and these red strands. This complementary DNA then hybridizes with the samples and eventually can be scanned because we are using fluorescence here. Now coming towards the main topic. Here I have taken a rather modified type of microarray technology that incorporates polymer in it. The polymer can be any species depending on the type of experiment, and it is being immobilized on chips. Um, Not I have used the term immobilized here because we can say immobilized is specific asset of microarray technology. And we are already familiar with stem cells, especially hematopoietic stem cells and mesenchymal stem cells, and their significant potential in regenerative medicine. Now, researchers have observed that when stem cells are allowed to come in contact with the polymer substrate, these are the mesenchymal stem cells and these are the polymer, some of the polymer substrates used here. Whenever they come in contact with polymer substrates, they elicit significant differentiation and regenerative abilities. That's why polymer microarray technology is of particular interest. Now let's see how these polymer microarrays are fabricated. There are two ways to design them. First one is liquid dispensing systems that allows for the deposition of monomer or polymer solutions onto substrates, forming microarrays with different chemistries. Contact printing and non-contact printing are two methods used to create microarrays. The second one is microfabrication based techniques. Um, and it utilizes techniques such as electron beam lithography and nano imprinting. Now, we'll see effects of chemical structures of a variety of polymer substrates on stem cells. Four types exist for this purpose. We'll discuss them one by one. First them is synthetic polymer microarray that incorporates UV or ozone radiation modified polystyrene culture systems in order to promote self-renewal. Now, both second and third types incorporate proteins as substrates. The only difference is that ECM protein microarray, which stands for extracellular protein microarray, has proteins from extracellular matrix, while peptide microarray has small proteins rather than whole proteins, and it works somewhat like this. Suppose I have this surface of a chip, and to this chip we have immobilized our peptide. I have shown it here by red, and whenever any kind of antigen specific for this immobilized peptide comes in contact, which I have shown as blue, it is identified and gets attached. Now, secondary antibody labeled as green recognizes this antigen, and this secondary antibody is being labeled, and this biomarker is shown as black portion here, and that's how it works. Now, the fourth type named small molecule microarray mimics the native extracellular matrix and eventually enable differentiation of stem cells. Now, we'll discuss effects of physical properties. Two properties are of particular interest, namely topography and stiffness of substrates. When it comes to topography, researchers have found that stem cells show positive response to integrin receptors. Integrins are like little hands that help cells grab onto surrounding environment. And as for the stiffness of substrates, they have negative impacts. So scientists use modified PEG hydrogels. PEG here stands for polystyrene glycol hydrogels to prolong muscle stem cell survival. Now, in order to simultaneously test the effects of chemical and physical cues, researchers have developed 3D hydrogel microarrays that have three-dimensional culture environments to understand stem cell behavior in vivo environment. If I'm saying in vivo environment, then it means it's environment that resembles somewhat like the inside of the organism's body. Now, the figure here shows fabrication of 3D hydrogel microarrays. It illustrates how a robotic workstation allocates composition on a microarray substrate. That's a robotic workstation and that's a microarray substrate. Next, polymerization of this hydrogel 3D element with probes is done by using UV light. And these blue droplets here represent 3D hydrogel elements along with probes. Now, this fabrication aids in testing matrix elasticity, proteolytic destability, and signaling proteins on mesenchymal stem cells, self-renewal, and osteogenic differentiation. Now, coming towards the perspective and conclusion of this technique, we can say polymer array technology is crucial for studying material properties and stem cell responses. However, it requires high throughput materials discovery, heat optimization, and mathematical modeling. Now, what I mean by this terminology high throughput here is that testing a large number of samples or compounds, sometimes even a whole genome of organism in short period of time. So that's really big deal. So that's like a challenge that comes in the way of this technology. But on the positive note, we can say high throughput microarray technology can help discover chemically defined culture substrates for hematopoietic stem cell culture. And with the advances in robotic liquid handling technology, hydrogel chemistry, and photolithography, development of three-dimensional scaffolds could accelerate, and the dream to utilize the stem cells for regenerative purposes can finally be realized. And that's all